Hi. Okay. თუ რამე adjustment ამჭირდება, იკავნა აქედან აუწე. Okay. Hello guys, how are you? Good. Okay. Now? Yeah, okay, great. So, let's begin. Actually, this should shut the box. How many should this pull to the box? Okay. I put. Alright, давай. Make sure you are coming. იდეაში არ არის პრობლემა. ეს არ მუშაობს, ხო?
ეს მუშაობს. აი, კამერა.
Okay, so guys, today we are going to discuss some effects of CD8 T cells, also known as effector T cells and cytotoxic T cells. So remember from our previous lectures we discussed T helper subtypes of CD4 T cells and effector T cells, okay? So do you remember why they were important, like T helper cells, and how many subtypes do we have? Just brief revision. Yeah, and also T regulatory cells, okay? But yeah, okay, we'll talk about this later. So now we need to discuss some CD8 effector T cell function. So do you see this well, or you need to like turn off? Okay, great. So as you already know, those CD8 T cells are involved in terms of elimination of some mostly viruses, but sometimes also they are involved in terms of elimination of some bacteria, like T helper cells. And also you probably started like studying some viral infections, and you know that the viruses can also infect those immune cells, but also non-immune cells, like hepatitis virus can infect liver cells. And now there is some problem because those T cells and some antigen presenting cells should degrade those viral containing cells and present these antigens to those T cells in order to activate T cells. And sometimes this leads to destruction of our normal viral containing cells, and that's why actually we get sometimes from hepatitis virus, we sometimes get cirrhosis and destruction of the liver. This is not due to the cytopathic effect of itself virus. This is due to our immune system destroys the viral containing cells, okay? So we'll discuss this too. And yeah, and sometimes even if the viral containing viruses can be in phagocytes, sometimes they express some ligands and some proteins that help them to evade the immune system, okay? And there will be some problem because those T cells can't even recognize those cells and can't destroy intracellular viral organisms, and therefore we will have a really bad infection, okay? And other than this, also this is CD8 T cells help to avoid some bacterial infections, and also they directly kill tumor cells along with NK cells from innate immune system, okay? And also we will study later that those cytotoxic cytotoxic T cells are involved in acute and sometimes in chronic graft rejection after transplantation. So that's why they are important, and that's why sometimes we use some medications that decrease immune system, like steroids, most commonly used, to actually avoid those acute graft rejection. Okay. Now we need, as you know, from the beginning and from the development of T cells, we have naive T cells which are actually unexposed to anything, and from those naive T cells we need to differentiate them into effector CD8 T cells. So there can be two ways, like you can differentiate into the CD4 T helper cells or into the CD8 T cytotoxic cells, depending on the antigen. Okay, and the antigen and the cells actually which contains those viruses, they are called target cells, and those target cells are actually destroyed by those T cytotoxic cells once they get activated. Okay, and this differentiation needs antigen presentation from antigen presenting cells, and also some growth factors like T-beta that lead to expression of some cytokines like IL-2, which is important for cytotoxic T cell development, and eventually when we have developed like T cytotoxic cell or CD8 cell, this will go to the specific inflammation area to those where those viruses are, and they will actually release some granules where cytotoxic like proteins or molecules like perforins and enzymes that actually cause the destruction of virus containing cells. Okay, and actually sometimes we have some problems with the perforin itself or enzymes, and that will lead to so sometimes will lead to immunodeficiency and autoimmune disorders. So we'll discuss this also. And also oh my bad, give me a minute. And during this killing process we need to know that only target cells are destroyed, because along with like target cells in the neighborhood there are normal cells which don't contain those viruses, and there is a precise mechanism by how those T cells eliminate only target cells, viral containing cells, and actually by this we are avoiding to damage by some to the normal cells. Okay, and also that interferon gamma is the main cytokine of T helper type 1 CD4 cells, but also these T cells produce interferon gamma in order to also activate. As you know, interferon gamma is a main factor for macrophage activation, and to increase macrophage phagolysosome function and degradation of some microbes. And also T cells produce this interferon gamma in order to activate phagocytes actually. And as I told you, there are some gene transcription factors like T-beta or L-mesodermin which actually promote the differentiation of naive T cells into the effector T cells like CD8 T cells, and they increase the production of some cytokines, those actually perforins, enzymes, which destroy those virus containing cells. So as you can see here, there are two pathways like T-beta and L-mesodermin pathway, and we sometimes need memory T cells and also need actually active cytotoxic T cells. So both can can be produced by the activation of CD8 T cells. And from the beginning, first thing, we need to present some antigen to those naive T cells in the lymph nodes or other lymph node organs. And after the presentation, there's the same mechanism like CD8 activation, like co-stimulation and things like that. Okay, this is the same mechanism like it happens in terms of activation of T helper cells. And after antigen recognition, those T cells will develop and will differentiate, will produce itself many like antigen specific T cells. There will be many, like this is called clonal expansion, so many types of antigen specific CD8 T cells are produced in order to like timely avoid microbial or viral dissemination. Okay, and also those T cells will go into the specific inflammatory or viral containing areas, and they will do their function to kill the target cells. It may be tumor cells, may be viral containing cells, depending on the antigen. So this needs like T helper activation, and firstly we need antigen recognition, as you can see here, and second signals. And also there is some immune feature called cross presentation, because sometimes when virus infect normal cells which are not immune cells, we need to present somehow those antigens, because normal cells, like roughly speaking, are not antigen presenting cells. They have MHC1, but they need some activation and some immune cells in order to present those antigens to T cells. And now mostly we need dendritic cells, which are main activators of CD8 cytotoxic T cells. And also sometimes other than dendritic T cells, we need CD4 T helper cells to activate CD8 cytotoxic T cells. And you understand this because the cytokine produced by T helper type 1 main is interferon gamma, but other is interleukin-2, and as you know, interleukin-2 also leads to activation of cytotoxic T cell. Okay, so that's why sometimes we need help from the CD4 T helper cells in order to fully activate and develop antigen specific T cells. Okay, so now the main cell is the dendritic cell, and firstly, after recognition of antigen by the dendritic cell, it will produce the peptides, which are actually antigens, and present on the MHC1. MHC1 is recognized by the CD8 T cell. Okay, so now we need here MHC1, and MHC2 is for the CD4 T cell. And also you should know that those antigens are presented on MHC1, they must be in cytosol, okay, in the cell. So they are called also intracellular antigens. And also for the comparison of CD4 cell activation, they are mostly presented, of course, by MHC2, and also those antigens are extracellular, coming from the extracellular area, okay, like extracellular bacteria, so some antigens. And after antigen recognition, after antigen is in cytosol or virus, they must be degraded by proteasomes proteasomal degradation pathway, which is, ubiquitination of with some, bacterial or viral proteins that will lead to degradation by proteasomes. And after proteasomal degradation, there is a normal immune response causing peptides or antigens which will be displayed on MHC1, and this process is mediated by TAP proteins, also known as transporter of antigen proteins, and they are important in terms of delivering the antigens from the cytosol to the outer surface of the cell. Okay, so T cells can't go into the some cells to actually recognize antigens. It must be presented on the surface by the MHC1. So the antigen is transported by those top proteins to display on MHC1, and this will lead to activation of T cells. And for the comparison, mostly MHC type 1 pathway involves intracellular pathogens in the cytosol antigens, and also CD4 and MHC2 presents to the surface the extracellular pathogens like many bacteria, and this is another pathway. Okay, so yeah, do you have any questions? Yeah. Okay, so let me show you a picture. So mostly there is a difference between MHC1 and MHC2 pathway. The presentation actually, type 1 involves proteasomal degradation, and type 2 involves phagolysosomal degradation of the bacteria, and therefore the result is the protein, which is then displayed on the MHC2. So there is a phagolysosomal degradation of bacteria. And also there is a basic difference between MHC1 and 2 in terms of structure. Have you studied this in your previous life science tree? Yeah. Do you remember the basic differences? So mostly MHC type 1 contains beta-2 microglobulin, which is not in MHC2, and also MHC2 contains invariant chain, so which actually blocks the antigen presentation, but normally it actually blocks the function of MHC2, but after there is some antigen in the cell, the invariant chain will be destroyed, so will come out from its blocking area, and on the MHC2 there will be presentation of antigens on the surface that will be recognized by CD4 T cells. Any questions? Okay. So and sometimes as I told you, some viruses does not infect actually the dendritic cells, which are mostly important in terms of activation of T cells, and therefore now we need some cross presentation. So I will discuss this now. And cross presentation means there is some activation of normal MHC1 cells, containing cells. So as you know, MHC1 cells are almost all nucleated cells. So basically they can present somehow some antigens like normal every nucleated cell, but it needs some help from the dendritic cells in order to fully activate CD8 cytotoxic T cells. And also there is co-stimulation important in terms of fully activation of CD8 T cells. And as you can see here, like difference between MHC1 and 2 presentation, there's a for the MHC2 there's a phagosome or phagolysosomal degradation of extracellular bacteria, and those peptides will be transported on the MHC2 surface in order to present to CD4 T cells. And for the antigen also intracellular or cytosol antigens, they will be degraded by proteasomal pathway, and after production of antigen peptides they will be transported by tau proteins on the surface, and this will lead to recognition of those MHC type 1 which has antigen on its surface. They will be recognized by CD8 T cells and TCR. So T-cell receptor recognizes MHC1 and also co-receptor is CD8. And do not confuse between co-receptor and co-stimulation. So co-receptor for TCR is CD8 and co-stimulator is like B7 and CD28. Okay. And also, as I told you, sometimes we have latent viral infections, or some cancers also lead to need in order to be eliminated from our cells, they need to activate T cells, and the mechanism is firstly we need to activate CD4 T helper cells in order to, like, they will help CD8 T cell activation. So in terms of latent viral infections or in organ transplant, we have firstly CD4 activation and then CD8 in order to produce very highly antigen specific CD8 T cells. Okay. And yeah, and also sometimes we develop some infections, mostly bacterial but also viral infections, and even people with HIV and AIDS develop some cancers. Why is that? Because as you know, HIV infects mostly CD4 T cells, and on a later stage it causes the decrease the levels of CD4, and like mostly the AIDS developed after several years of the infection, and AIDS means there's a less than 200 CD4 T cells, and this is a problem because those CD4 T cells can help activation of CD8 T cells, and therefore, as you know, CD8 T cells eliminate tumor cells, and we will have some increased proliferation of tumor cells, and therefore people with HIV and AIDS develop very severe infections and very severe and fast proliferating tumors like HPV, which can lead to early transformation of, as you know, you can detect from pap smear this HPV and can lead to cervical cancer, and also sometimes anal cancer can be caused, and also head and neck cancer, and many types of cancer are caused by those HPV, but also Epstein-Barr virus may cause many cancers. So that's why we need CD4 T cells to activate CD8 cytotoxic T cells. So only dendritic cells are not main and are not important in terms of activating CD8 T cells. So you need to understand why we have some opportunistic infections in AIDS. Pathogens? So only dendritic cells are not important to activate CD8 T cells, but not only. Yeah, so they are most important, but in terms of latent viral infections, tumors, now we need CD4 to activate, okay? Yeah, so organ transplant, tumors, latent, they are most important, like dendritic in normal acute viral infections, but in terms of tumors and latent viral infections, we need CD4 T cells. Like, do you know any latent viral infection? Virus can cause latency and therefore develop some latent viral infections. Yeah. Yeah, so they actually reside in some ganglia in our cells, and therefore sometimes may lead to flare up of herpes or zoster or anything, and also Epstein-Barr virus, also with a type of herpes virus which can lead to development of latent viral infections. And now that's why we need CD4 T cells to for the activation of CD8 cytotoxic T cells in order to destroy them. Okay. So, and how these CD4 T cells activate CD8 T cells? So we have some pathways by which there is activation of CD8 T cells. So firstly, it secretes cytokines like IL-2, which is one of the most important in terms of activation of CD8 cytotoxic T cells. Also, activated T helper cells express some CD4, which is also important for CD8 cell activation, and also there is an expression of co-stimulators like B7, and all this leads to activation of T cells by CD8 4 cells, and IL-2, which is produced by the CD8 T cells and also by the T helper type 1 cells. They lead to they promote actually proliferation of CD8 antigen specific cells, and therefore we have a full activation of CD8 cells, and also it leads to differentiation of activated CD8 cells into the memory T cells. Okay, we have also memory CD4 and CD8. Okay, in order to, after another time when there is an invasion of same virus, we need to have antigen specific memory T cells in order to actually kill those viruses very fast. And there is some mechanism by which IL-2 leads to the activation of CD8 T cells. It leads to expression of mostly alpha chain is highly expressed. So as you know, there is some disorder when we have IL-2 gamma chain problem, IL-2 receptor gamma chain problem. Do you remember? Sorry? No. So yeah, this is one of the form of a severe combined immune deficiency. So IL-2 receptor has several, structure which contains alpha, beta, and also gamma chain. So both three are important, and this IL-2 leads to expression of alpha chain and also other chains, and this will lead to IL-2 will bind to these T cells and will lead to differentiate into effector T cells. And that's why sometimes when we have IL-2 receptor gamma chain mutation, or when we have very low levels of this chain, there will be a combined immune deficiency, means T T cells and also B cells are not activated well, okay? And this will lead to many viral infections and also bacterial too. So other than interleukin-2, also interleukin-12 mainly is produced by macrophage, but also CD8 cytotoxic T cells, and along with some interferon alpha and betas, they also lead to differentiation of T cells into the effector CD8 or CTLs means cytotoxic T lymphocytes, also CD8. And also other than IL-12 and 2, we have interleukin-15, which is also important for the survival and the development of memory T cells. And also IL-21 is important for the activation and production of effector and both memory T cells. But mainly, like if you don't memorize those cytokines, mainly you need to know IL-2 and also interferons, which are the important in terms of differentiation. So here you can see we have CD4 T helper cells that produce cytokines like 2, 12, 15 that will lead to activation and expression of some co-stimulators and there will be recognition of antigen by T cell and really good activation of CD8 T cells, which will differentiate into either cytotoxic CD8 T cells or into the memory T cells, depending on the antigen. But actually there is no antigen dependence on this because we need both now active cytotoxic T cells and for later we need memory T cells. So there's actually both are differentiated. And also other than cytokines, CD4 T cells enhance the ability of antigen presenting cells to present more antigens to T cells, and there's an expression of CD4, which is important for the CD8 activation, and therefore you also remember the disorder which is caused by CD4 mutation, which can cause T cell dysfunction. Do you remember CD4? And this CD4 is also important for B cell activation and class switching of immunoglobulins. So we had only immunoglobulin M production. Yeah, so this is the same. So this will lead to decrease in all immunoglobulins other than immunoglobulin M because we don't have class switching, and also we have some defect in T cell activation. So there will be viral infections and also bacterial infections, okay? Because we don't have activation of cytotoxic T cells and also we don't have memory T cells. And sometimes, we have when we have some chronic infections, mostly viral infections like hepatitis C, B, and also Epstein-Barr, other like herpes viral infections, those viruses express some proteins that lead to decrease the activity of T cells. Like remember we discussed PD-L1 and CTLA4, like some tumors express those proteins that lead to decreased activation of CD8 T cells. So this is the same mechanism, and this is called T cell exhaustion because T cells can no longer destroy viral cells or tumor cells. And and this is different from immune tolerance, because in tolerance, like lymphocytes actually fail to develop into the effector T cells, but in terms of exhaustion we have initial activation of T cells, but then there is a shutdown of immune response. Any questions from this? Okay, so now we'll discuss what proteins and what factors they express in order to downregulate immune system. So mostly like tumor cells express, they're the kind of same mechanism, but they're a bit different. Mostly they express PD-1 or PD-L1, which is ligand for PD-1, and also CTLA-4, and CTLA-4 inhibits like immune response. So those viruses try to express those proteins on their surface in order to avoid T cell activation, and they will proliferate and will rise to develop severe infections. And also LAT3 is another important for T cell activation, and actually inhibits T cell activation, and those viruses express this LAT3 and CTLA-4 in order to avoid immune system. And why do we need this? As you know, like let's talk about HCV. So HCV actually does not kill hepatic cells. Okay, they are the immune system CD8 T cells which destroy those normal liver cells. So this is kind of mechanism by which chronic infections, by their expression, they lead to less activation of T cells in order to avoid severe damage of the liver. Okay, this is kind of cohabitation, okay, in order to avoid severe infection. But sometimes this is not enough for the controlling of the infection, because if we don't have some destruction of viral containing cells, the virus itself will proliferate, and again they are like HCV is not very cytopathic, so can't actually severely damage cells, but it can by some level. But therefore this is a mechanism that helps to avoid severe infection. But again, this is not enough for the immune response and for the chronic infection. Yeah. So you mean why we have this response? So I think like there are not good research and good at how they are developed, but I think like tuberculosis, we like normal our cells produce granuloma around those bacteria, because tuberculosis can't be eliminated by those CD4 T cells we have discussed previously. So this is kind of same mechanism by which there is an in the liver, like in terms of HCV, we have some development of scars and collagen around those viruses, and therefore it actually contains those viruses in order to avoid the dissemination. But this is not enough. And why do we have this? Because if we have severe activation of T cells, they will destroy those viral containing hepatic cells, and therefore we have some decreased immune response. Okay, here we got it. So if we have severe activation of T cells, cytotoxic T cells, they will damage and destroy our liver cells. So therefore we have some response that leads to decreased activation of these cytotoxic T cells, and therefore there's a delayed, damage of hepatic cells. So eventually, if we don't give the medication to those patients, there will be infection and eventually to lead to cirrhosis and sometimes hepatic cancer. But this is kind of delaying the response, because those T cells, if they are very active, they will lead to severe destruction, and the infection will lead to its peak at a very, very early stage. Okay, so this is kind of avoiding, but actually not enough. And okay, so here you can see normally when there's acute infection, those T cells are activated, CD8 T cells, and they will respond by proliferation, differentiation into antigen-specific T cells, and this will lead to target killing. So target means any viral containing cells will be killed, but some viruses express other proteins that lead to evasion of the immune system, and therefore we will have some chronic infection. And in terms of chronic infection, to avoid more T cell activation, and so any immune cell activation leads to tissue damage and inflammation. So inflammation, over-inflammation is not good. So therefore those T cells are exhausted and downregulated in order to avoid over-inflammation and more tissue damage, like in liver in terms of HCV on HPV, and also like for Epstein-Barr virus, it can lead to, in terms of chronic infection, some lymphoproliferative disorders, because Epstein-Barr virus goes into B cells by CD21 receptor, and in terms of chronic infection, it can lead to development of some lymphoproliferative disorders and some lymphomas. Yeah. Oh. Okay. Take a break. Okay, let's take a break, guys. Okay, let's continue. So we have so much to discuss now. We have discussed the exhaustion of T cells, and not only viral infections lead to exhaustion of T cells. Like sometimes we have tumors that express cells like PD-L1, CTLA4, or LAG3 that leads to exhaustion and downregulation of T cell activation, and therefore we had some medications that targeted those proteins like PD-1, PD-L1, and that will reverse the T cell exhaustion. Okay, so that will activate T cell in order to kill those tumor cells. And also we have some medications for LAG3 that is combined with PD-1 nivolumab, which and most of these medications are used in many types of tumors like melanoma, kidney cancer, lung cancer, and many types of cancers, because many cancers express those proteins on their surface in order to avoid T cell activation and destruction of tumor cells. Yeah, so not only viral cells express those proteins that lead to exhaustion of T cells. But hopefully we have some medications that reverse this exhaustion of T cells and will activate T cells and destroy those tumors. So additionally, interferon gamma is also produced by T cell, which as this leads to classical macrophage activation pathway also. So when we have any kind of infection, you don't try you should always think that there is no only one diversity and only one activation of T helper type 1 or only activation of macrophage. We have some kind of combined activation of also macrophage, neutrophils, T helpers, T cytotoxic cells. Okay, there's a combined immune response and not only only one. Okay? And therefore when we have T cell activation and CD8 T cell along with sometimes T helper type 1 CD4 cells, they will release interferon gamma, and macrophage has increased activity of its phagolysosomal function and therefore more robust and more destruction of sometimes viral containing cells, sometimes bacteria. Okay. And also we discussed this that there is some mechanism by which T cell destroys those target cells or viral containing cells. So there will be a specific bond between T cells and target cells. This is called immune synapse. Like we have neuronal synapse or any synapse in our system, because this mechanism needs to be very precise in order to avoid damage to another neighborhood normal cells. Okay, so there will be a synapse between those T cells and target cells, and there's an increased expression of some integrins like LFA1 on T cells or lymphocyte functional antigen type 1, and also there's an increased expression of ICAM or intracellular antigen molecule on those target cells, and this will lead to strong bond between T cells and target cells, and therefore immune synapse is formed. And after the formation of immune synapse, there is a mechanism and I told you in the beginning that those T cells destroy the target cells by releasing some granules where are like perforins, enzymes. So after this immune synapse formation, these granules are exocytosed from the cell and they will go directly to the target cells and not to the like space. Okay, and after this granule exocytosis, there will be detachment of T cells because T cell already recognize those target cells, so we need to find another those antigens in other cells. Okay, so after detachment of T cytotoxic cells, there will be a target cell death by those enzymes and by those proteins like perforins and coenzymes, which actually both of them mostly coenzymes lead to activation of programmed cell death pathway or apoptosis pathway, okay, or death. And there are involved some enzymes called proteins called caspasas, you may know from your pathology, which is very important in terms of apoptosis. And also like there's an expression of some integrins like leukocyte function associated antigen and also ICAMs, which will lead to formation of the immune synapse, which is important for precise killing of target cells and not other cells. Okay, so after we have some activation, and they will kill all like any nucleated cell which have those antigens, those viral antigens, okay, and no other cells should not be harmed. But sometimes we have some problems with those sometimes cytokines or even proteins that will lead to many T cell activation and destruction of other normal cells. Okay, like we have discussed previously, toxic shock syndrome from Staphylococcus. There is a specific superantigen that leads to too many activation of T helper cells, which releases many types of cytokines like T helper, like IL-1, TNF alpha, interleukin 6, and all this will lead to development of shock and severe, infection. So that's why we need precise killing of any nucleated cell which contains those antigens. So therefore they are called antigen specific T cells. Okay. And there's a mechanism like those T cells contain some granules or like lysosomal granules that are secreted from lysosomes, and like also neutrophils have some granules, and same is for the T cells. And in these granules we have those coenzymes and perforins which will lead to the killing of those viruses. And other than this, like after immune synapse formation and after those integrins expression, and when there's a tight bond between the T cells and target cells, there will be exocytosis of those granules in the outer surface and into the between those target cells and T cells. And after exocytosis, there will be actually delivery of perforins and coenzymes into those target cells, and that will lead to activation of apoptosis pathways. So here you can see the caspasas are proteins also known as cysteine aspartate proteases. There are many types of caspasas, but mostly they are involved in terms of cell death activation or apoptosis activation, And also we have mainly two proteins which are involved in this. Firstly, in those granules there are perforins which actually perforate and development of hole or pores by which there is a transportation of granzymes into the cell. So perforin makes holes, therefore granzyme will go into the target cells and activate those pathways. Any questions regarding this? Sorry? So glycine. So sometimes we actually don't have antigens and antigen-containing cells. We don't need those granzymes to be released from those T cells. So this glycine holds those enzymes into inactive states, and after T cell activation and cytotoxic T cell development, there won't be any glycine. Therefore, those granzymes will be released into the immune synapse, and therefore this will lead to destruction of viral containing cells. So that's why glycine is important. And also, as you may remember, C9 complement protein from your previous maybe lecture. So do you remember membrane attack complex of complement? So complement activation, the last stage is to produce membrane attack complex to destroy some bacteria. And those there are many proteins like beginning from C5, C6, 8, and 9. All those are called membrane attack complex, which lead to development of holes into the bacteria. So the perforin has the same function, to produce some holes, and therefore granzymes will get into the cells. So you should also know what is C9. And sometimes we have problem with the formation of later complement proteins like beginning from C5, C6, 7, 8 and 9, and you may know there is some infection which those patients may develop. Do you remember? Like when we have C9 or C5 or membrane attack complex problem, do you remember any infection that can be very severe in those patients? Have you studied gram-negative bacteria like Neisseria? Yeah. So when we have those C9 complement deficiency, or beginning from the C5 to C9, or also known as membrane attack complex, there will be a predisposition to severe infections caused by Neisseria meningitidis, and which we already know it can cause some severe meningitis, also Waterhouse-Friderichsen syndrome, which is the hemorrhage into the adrenal glands, and severe adrenal insufficiency, and also those patients along with meningitis have typical rash on the surface. So we should know that those patients must be vaccinated against Neisseria infections. So that's why this C9 is important, and also perforin. But now this perforin does nothing has to do with this C9. This is just for the comparison of perforin is from adaptive immune system, from developed and released from CD8 T cells, and C9 is from innate immune system like complement protein. And as we discussed, perforin forms pores into the cells, into the target cells, and therefore granzymes will be delivered. This will lead to activation of caspases like mostly 3, which are important for apoptosis promotion. And also there is a activation of pro-apoptotic protein like PID. This is also pro-apoptotic, and this will lead to activation and trigger the pathway of apoptosis. And also, other than this granzyme, so please differentiate granzyme from granulosin. So granzyme is another protein which activate caspases, but also we have granulosin. This, along with perforin, can alter the permeability of cells, target cells, and also can help the perforins to produce pores into the cells. So granulosin is for this. Okay, so other than granule or those perforin and granzyme dependent pathways, we also have other pathways that lead to activation of apoptosis. So another one is those T cytotoxic cells express Fas ligand, and those Fas ligand binds to death receptor, also known as Fas. And this is very important in terms of non-granule dependent activation of apoptosis. And so why is this important? Let me tell you in a minute. So firstly, we have granule dependent pathway, which is mainly mediated by perforin and granzymes, which will lead to exocytosis of those granules and deliver to those target cells and activation of apoptosis and apoptosis of target cells. And other Fas or Fas ligand pathway, which is granule independent. So sometimes, so why we need this Fas and Fasel pathway? Sometimes when we have normal, this is not only for target cell elimination. Also when we have normal immune response, and after those immune response there are T cells in our cells, and sometimes those T cells must be eliminated after antigen elimination, okay, because we don't need those T cells to be too much in our organism, and therefore this apoptosis pathway is also important for T cell elimination itself, okay. So in those T cells they express Fas ligand, and they will lead to death of itself, those T cells, in order to avoid too much cytotoxic T cells in our system. And there's an important correlation. When we have some problem with Fas ligand or Fas, this will lead to defective apoptosis of those T cells, and now we would have some overactive T cells in our bloodstream and in our system. So this will lead to autoimmune disorder, also known as autoimmune lymphoproliferative syndrome, because this mechanism is kind of similar like negative selection in the medulla. So do you remember what is negative selection and why do we need this? Any ideas? Negative selection in the thymus during the early development of the T cells. Yeah, kinda. So we need negative selection in order to eliminate highly reactive T cells, and they must not be released into the bloodstream. But this is the same mechanism when we have problem in the periphery and problem with the destruction of reactive T cells, that is caused by Fas or Fas ligand interaction problem. We will have overactive T cells in our bloodstream. and this will lead to severe autoimmune disorder called autoimmune lymphoproliferative syndrome, which will lead to severe liver and spleen enlargement. Also, pancytopenias. Do you know what the pancytopenia is? So pancytopenia means decrease in all types of blood cells, like decrease in neutrophils called neutropenia, decrease in red blood cells called anemia, and decrease in platelets, thrombocytopenia. All these combined together means pancytopenia. So pan means all of them is decreased, and cytopenia means cells are decreased. So pancytopenia is very severe disorder, which may cause many other reasons also, but this will lead to development of infections due to decrease in leukocytes and neutrophils. This will lead to bleeding because of the decrease of platelets, and this will lead to anemia because of the decrease in red blood cells. So this is pancytopenia. And autoimmune means these are T cells actually which lead to destruction of those some other T cells, red blood cells, platelets, and others. So okay, this is like simplified version. So initially we have CD8 T cell activation and recognition of MHC1, and after this activation T cell will release granules which contains perforin and granzymes, which will produce pores into the target cells, and therefore granzymes will lead to activation of this apoptosis and lead to the destruction. And as you can see here, there are also like several pathways. One mediated by cytotoxic T cell granule dependent pathway. On the right side you can see perforin and granzyme B, which is most important in caspase activation. And also we have fast ligand and fast interaction, which is granule independent pathway. Both leads to apoptosis and cell death. Okay, any questions? Okay, let's continue. So now also we talked about interferon gamma, which is also important cytokine for CD8 T cell, and this leads to activate of classical phagocyte pathway and elimination of some ingested microbes, because sometimes we don't have like alone viral infections. There may be like in our system combination of some infections and invasion of some types of bacteria and viruses simultaneously. So therefore we need to have ready, and we need to have activate those T cells and also macrophages. Okay. So now sometimes we have talked about this, that those many viruses express some proteins on their surface that lead to evasion of immune response. So even if they are in our immune cells, like Epstein-Barr virus into B cells, sometimes even those viruses can't be eliminated because they express some proteins that lead to decreased activation of T cells mostly, and sometimes NK cells. So therefore we will have chronic viral infections, and this is very important how those viruses and also bacteria lead to development of chronic infections. And other than viral infections, we have chronic like mycobacterial infections like tuberculosis, which leads to granuloma formation and confinement of bacteria in one place. And other than mycobacterium, there are like listeria, which are also intracellular bacteria, and all this leads to expression of some proteins on the surface in order to avoid immune system response, and therefore we will have chronic viral infections. So there are several mechanisms by which they decrease immune response. So firstly, this is very important for tuberculosis because it contains sulfatides, which are glycolipids. Have you covered TB already, mycobacterial infection? So keep that in your mind because this is also very hard for your board exams. Sorry? I just asked you if you have studied with TB. These are infections that bacteria or viruses that express some proteins on their surface to avoid immune system activation. Yeah. So mostly mycobacteria have sulfatides, and even if they are in phagocytes, those sulfatides actually prevent phagolysosome fusion, and therefore we will have defective phagolysosomal fusion and therefore defective elimination of mycobacteria. But for now, for viral infections, which where CD8 T cells are important, they also express some proteins or lead to the defective HSV or herpes viruses lead to defective transportation of peptides on the MHC surface. So therefore we will have we won't have actually T cell activation and therefore latent chronic infection. So this there may be the same for cytomegalovirus and also Epstein-Barr virus. They use different mechanisms. And also, other than this, Epstein-Barr virus also releases interleukin-10, which is important for decreasing the immune response, and therefore chronic proliferation of T cells, decreased proliferation of T cells. Okay. We have discussed these infections like hepatitis B or C viruses. They actually don't destroy the liver cells. There are T cells which causing liver damage. So you should also know this correlation, and also other than these protective roles from CD8 T cells, they may also lead to acute or chronic graft reactions in transplanted patients. So that's why I use some immunosuppressants that actually decrease T cell activation and therefore prevention of graft rejection. And also, as you can see down there, sometimes, and we already discussed one disorder when we have overactivation of T cells, but there is also important correlation which is also in your book, but I would like you to tell you more details. When we have severe and defective activation, defective I mean very robust activation of T cells, it will release too many interferon gamma, and those interferon gamma will lead to activation of many macrophages, and normally we don't need those macrophages to get activated like too many. So therefore there's a disorder called hemophagocytic lymphohistiocytosis, which means hemophagocytic means hemo is blood, and phagocytosis means the destruction of blood cells and decrease of blood cells. This will lead to pancytopenia also. And lymphohistiocytosis means there's an overactivation of macrophages by those interferon gamma, and therefore this is very problematic because and mostly there are many causes of this HLH shortly, this disorder, and one of the main reason is in congenital forms there's a defective perforin, and this defective perforin and this mutation leads to overactivation of T cells, and normally like those T cells are activated but the perforin can't do its function, so can't kill those target cells, but when we and therefore we have more and more activation of T cells in order to overcome this, but it does not help. Okay, it leads to many interferon gamma release and excessive macrophage activation, and that's why we have this vicious cycle that leads to tissue damage and the development of the disorder. So other than these congenital forms of perforin mutation, we also have acquired forms of this disorder, and this means some infections, mostly viral, like Epstein-Barr virus, can also promote this defective and overactivation of T cells, which will lead to release of interferon gamma, and this will lead to development of clinical presentation of these disorders. And other than interferon gamma, interferon gamma is main activating factor for macrophage, but you also know macrophage also releases many cytokines like interleukin-1, TNF, interleukin-6, and all this leads to activation of many macrophages, and also sometimes we have other cytokines that lead to CD8 T cell activation, and therefore we have tissue damage. And here you can see that overactivation of those macrophages and cytotoxic T cells will lead to development of fever, because fever is mediated by many cytokines like interleukin-1, 6, TNF-alpha. So we will have severe reluctant fever, also pancytopenia or decrease in blood cell types, and also enlargement of the liver, and many other symptoms. And here on this picture you can see that there is a phagocytosis of normal blood cells. It may happen in the bone marrow, but also in the periphery. Okay, and this is just for you. So yeah. But mostly perforin is important along with granulyzing, but mostly it's perforin, so, and granzyme. So if you don't have perforin, this granulozyme is not alone capable of promoting pore formation. Okay, so mostly we need perforin, and this is just for you for clinical correlation. You can read all this at home, but there are many causes of this disorder, and like maybe triggered from viral infections, bacterial infections, maybe triggered by cancers also, and all this lead to defective activation of CD8 cells and macrophages, and eventually will lead to destruction of our normal cells. This is kind of autoimmune disorder and can be very severe. Okay, I think we finished Any questions? You look like you understood everything. Well, yeah? Or no? Yeah. What? Yeah. Yeah, this is not easy. I know. Yeah, I understand. You look like drowsy, sleeping. Yeah. Yeah, I understand. Yeah, but yeah, I understand your concern, but also you need to read the previous lectures and understand, others new lectures. Okay? What? Yeah, I can send you. I already sent this slide. Yeah. So, and that's why tutorial is important. So we are discussing the most important facts. Okay. Sorry. Yeah. Other than tutorials, you mean? Yeah, so I can also suggest you to do, you may know RX, this is a kind of question bank, or also Ambos question bank. Okay, I can send you all, yeah, for immunology, yeah, for sure. Okay, we have some time. You wanna rest or discuss something? Maybe you have questions from like previous lectures. Anything? Okay. Yeah, I think we finished. Okay.